Welcome to New Topics, New College. I'm Steve Miles. I'm the provost of New College, and we're delighted that you could come for this evening's talk. Most of you, I think, came in through the doors over there, but we're doing something new. So when you leave, you'll maybe want to leave out of those doors. We are doing something with a student this evening that I think you'll find very interesting. As you know, undergraduate research at New College is very important. All of our students complete a senior thesis. It's unlike many colleges where it's an option or it's an honors thesis. Here, every student completes a thesis. It's usually a year-long project, and many of the theses are published. They're really of master's level quality. So what we have for you this evening is you can sample a senior thesis on your way out. We have a student, Cody Gunton. He's a fourth-year student in math, and he'll be in the lobby. Again, he has a poster over there, and he'll be able to talk with you about his thesis. And I'm going to read what this is. It's studying pure math using mathematical structures of physics. He is working to construct the mathematical equivalent of a system of quantum mechanical particles with symmetries that arise in the study of the integers. That's a senior thesis at New College, okay? So I hope that after the presentation and after the Q&A, when you join us in the lobby, just look on the wall over outside of those doors, and Cody will be there, and I'm sure that he'll be delighted to tell you about his thesis. In fact, he probably has a bumper sticker, ask me about my thesis, that kind of thing. We are very thankful to our sponsors for New Topics, New College, the Herald Tribune Media Group, SNN 6, WEDU, Marriott, and Madison's have all been very generous this whole New Topics season. With us tonight is Mike Conley, the Executive Director of the Herald Tribune Media Group, who would like to say a few words. Mike.
Welcome from everybody at the Herald Tribune. I love New College, and I'm especially fond of this series. It is devoted to the same thing we are devoted to at the Herald Tribune, which is trying to make this a better, smarter, more well-informed community. I've been to some of these in the past. It's a great series. You're going to enjoy it. One piece of advice, I will tell you the same thing I tell reporters when they're walking out of the newsroom door. Ask hard questions. You get much more interesting answers. Enjoy.
Thanks, Mike. Thank you, Mike. Now, before we go further, I'd like to remind you about the next New Topics lecture in 2 weeks on March 20th. Dr. Paul Hansma, a proud New College alum, will discuss medicine atom by atom, from the atomic force microscope to the clinic. So again, that's March 20th, and we hope that we'll see you here. Before I introduce our speaker, I'd just like to say a few words about the relationship between New College and the Roskamp Institute. I think as most of you know, the Roskamp Institute is really close by. It's just up in Bradenton. It's really more like a bike ride from New College. But it's more than proximity that matters with us. We share a passion for research and for education. And for a number of years now, we have had New College students who are working in the labs at the Institute. And they share in the research with the folks at Roskamp, and their research is then translated into senior theses, for example, or independent study projects, tutorials, and so forth. That kind of real-world experience is essential, and to be able to work with scientists of the caliber at the Roskamp Institute is just a tremendous thing for us to have. So we're very, very pleased about that. Now, I know that for all of us here tonight, I would imagine that there's not a person in this room whose life has not in some way been touched by Alzheimer's. We either have a family member, a friend, friends of friends. All of us have seen the terrible destructiveness of the disease, and not just simply what it does to brain functioning, but the effect that it has on families. So I think we're all very, very eager to hear tonight, not only about the disease, but more importantly, some really great strides that are being made towards treatment. And we look forward to hearing from Michael. Dr. Michael Mullen is president and CEO of the Roskamp Institute. And I know you have a biography of him, some biographical information about Michael in your booklet. One of the things that you'll see is that he has about 5 degrees, and that's impressive enough, but I noticed that there seems to be one degree that might be missing here, where he has expertise. I was in the lobby talking with Michael, and he was talking about he'd just come back from Ireland, where he was working with scientists, and he said, you know, it's really quite a task. You have 300 scientists trying to get them organized. And what you could hear was the managerial skill that is required to bring projects like this to fruition. He is himself a great scientist, but it takes that special talent to be able to get people to work together, and certainly for this purpose, to find a treatment for Alzheimer's. We really appreciate everything that he is doing. So please welcome Dr. Michael Mullen.
Thank you very much, Steve. It's a real pleasure to be here this evening. My goal is going to be to bring you up to speed on what we now think causes Alzheimer's, and importantly, of course, what our efforts are to bring this rather disastrous disorder to an end. And I'm going to touch on several topics that are not just scientific. I'm actually going to talk about some things that we may be able to do ourselves to combat the disease and delay the disease. So here's the overview. Who's at risk for the disease? That's something that I think is of great interest to all of us. What is the disease? Exactly what is Alzheimer's? How does it work? Why does it destroy the brain in the way that it does? What do we know about that? What causes it? You know, what triggers it and makes the disease progress? And how can we as scientists stop it? And what are we doing to bring it to an end? And as citizens, what can we do to reduce our risk? Because I think one of the things that's changing, at least in my mind, over recent years, is that we're not entirely helpless against this disorder. There are quite a few things that are emerging from the literature that we can do on a personal level as we wait for new medications to come along to tackle this disease. But from my point of view as a physician and a scientist, of course, what I want to see are medications that can really bring this thing to a halt. And that's what I'll talk about in most detail. Necessarily, I won't be able to cover all topics, of course, and I certainly won't be able to cover all approaches. I will adopt a bias towards what we're doing at the Roskamp Institute. I think it's exciting enough. I think we have made great progress in advancing a drug, which I'll talk to you about, into phase 3 clinical trials, something I'm immensely proud of, because there are so few drugs in the world currently being tested. And you'll hear me say again and again that we need more drugs out there. We need to tackle this disorder much more vehemently than we currently are. Okay, so who's at risk? Well, the answer, as you know, is all of us. And why is that? Well, because we're living longer. And as you all are probably aware, Sarasota is a sort of a look-forward city for what America's going to look like in not too many decades' time. So the average percentage of individuals over the age of 65 is about 13%, something like that, in the United States as a whole. It's about 17 in Florida, but in Sarasota it's about 32%. So we have a much higher rate of individuals over the age of 65. The weather's nice, people come here to retire. But with it comes this bump in the risk for the disease. So we're looking at who will get the disease in 2020, 2030, 2040, and 2050. And these are the individuals in red over the age of 65, and you don't see much change there. It's not going to be the individuals in the lower age group that are really at risk. The ones that will increase in prevalence of AD, of Alzheimer's disease, are the 75 to 85-year-olds in green and the over 85-year-olds. And this bump here is the baby boomers aging. So you see this massive, and this is just in the US. So look here, we've got about 15 million people by 2050 that will have Alzheimer's. And these are the ones with diagnosed Alzheimer's. They're not the ones in the very early stages, which are probably in the order of 3 times more. So there's some debate in the field about when Alzheimer's is going to overtake cancer as the primary cause of death in this country. But everybody's agreed that it's not long. So this is a problem not just for us, but for all developing countries. Anywhere where individuals live beyond the age of 65, we are at the mercy of this disease. So here on the left is a healthy brain, and here on the right is a depiction of somebody with Alzheimer's disease in a fairly advanced stage. And what you can see are these big holes in the brain where there used to be brain tissue. The neurons in the brain, of which there may be as many as 100 billion for most of us, are systematically destroyed. The brain shrinks because the neurons in the brain get killed. And they get killed in the initial stages in particular areas. This area down here, or around here, is called the hippocampus. It's part of the temporal lobe. It's on the side of the brain. And that's the area that you are all using right now to take in new information. And when you absorb new information, it reverberates in neurons, in circuits in this part of the brain. And then over time, it gets transferred to the cortex. So long-term memory is stored in the cortex, all around the cortex, but short-term memory is stored in the hippocampus. And it's by necessity, the neurons in the hippocampus must be active, they must be alert. This is a very ongoing active process. It's not a passive process. The neurons have to be functioning and communicating with each other. But in Alzheimer's disease, you see this damage to the hippocampus. And so this is the reason why the first signs and symptoms that we see in Alzheimer's are of loss of the ability to recall recently presented information. That's why it's short-term memory loss. I use that term loosely, but the ability to recall a phone call from this morning or a visitor from the night before. Those things have to be processed in the hippocampus and transferred. When the hippocampus looks like this, that can't happen anymore. The information never gets transferred to long-term stores. So it isn't quite right that we forget in Alzheimer's disease. That's a little bit of a misunderstanding. What happens is we don't encode new information. And the other area that quickly becomes damaged are other parts of the temporal lobe and the parietal lobe up here, which deal with language. So again, one of the relatively early signs is the loss of ability to find words. Word finding problems are quite typical in the very early stages. Now, as the disease progresses further over time, then the rest of the cortex is finally destroyed. But it's not atypical for people in the early stages to remember events from 20 and 30 years ago much better than those that occurred recently. So what is happening? Well, this is what we believe is happening, and I'm necessarily going to take a slightly amyloidogenic tack. And let me explain what I mean by that. We are all making amyloid. It's a small peptide, which I'm going to talk quite a lot about. And we clear it. We make it in our brains, and we get rid of it rather quickly. But in Alzheimer's disease, it accumulates. And it accumulates in these clumps. And as a consequence of that, these neurons, these nerve cells that we use in a circuit to remember where we parked our car or what time we were supposed to be here or who else we came with, the neurons start to get damaged. While that is happening, as this amyloid accumulates, here's a healthy neuron, while it accumulates, the railway tracks inside the neuron, these things called microtubules, which are the really transporter systems that allow the transport of chemicals all the way down this branch of the neuron to the next neuron, those things start to become disrupted. And they're comprised of a protein called tau. So there's two main proteins involved, amyloid, which forms these diffuse aggregates, and this other protein called tau, which is really important for keeping this railway system together. It really holds it together. When amyloid accumulates, it starts to damage the neurons, and the tau falls apart and forms these structures called tangles. So I think I've got some neuropathology of that. Here's an actual Alzheimer brain, and here are neurons that are linked together in the way that I suggested. And here we have an accumulation of the protein called amyloid. And so this is a so-called senile plaque. And in the middle of it, this protein here is amyloid. Here's one with a very dense amyloid core, but there's actually another one here that you can't see so clearly. But nevertheless, the accumulation of this amyloid is very damaging to the surrounding neurons. And here's a tangle. You can see this occurs in contrast to amyloid, which accumulates outside of the cells. Tangles occur inside neurons. And once this happens to a neuron, once tangles accumulate, it's dysfunctional. It can't work anymore. It can't move neurotransmitter or other chemicals from the center to the end. It can't relate to other neurons. It can't send signals. So when you see this amount of damage or this amount of accumulation of tangle, you know that this cell is committed to dying. So this was a discovery that we made about 20 years ago now. It's actually a very well-known discovery all around the world because just about every pharmaceutical company in the world and many, many Alzheimer labs use this discovery. This was something that was actually found in Florida by myself and my colleague Fiona Crawford. And what this is a DNA sequence from two individuals, they're siblings. One of them is perfectly normal and has this sequence. And here are the 4 letters of DNA which go to comprise the genetic code. And the way you read these things, in those days anyway, was you read them as, going from bottom to top. So this actually reads AGG, T, AG, AA. That's the genetic code in that individual. And I can pretty much guarantee if we took a blood sample from all of you and we sequenced that part of the genetic code in all of you, that's the sequence that you would have. But look at this individual here. You can see that this individual has the same code here, although it's fainter. You can see that there's the same code. So here's the same 2 nucleotides here, and these 4 are these 4, but these 2 don't appear here. They just appear in this individual. So this is 2 DNA letters, if you like, misspelled out of 3,500 million. But nevertheless, the consequence of that misspelling is to give this individual Alzheimer's disease at the age of 52 or thereabouts. And the inheritance of this mutation confers an inescapable propensity to develop Alzheimer's. If you are unfortunate enough to inherit this mutation, you'll get Alzheimer's. Now, fortunately, it's extremely rare, but it was a very, very important finding because it was one of the first cues that we had that the accumulation of amyloid could cause Alzheimer's, because this error is occurring in the Alzheimer amyloid gene. It occurs in the amyloid gene, and that's gave us a very strong indication that the accumulation of amyloid alone can trigger the disease. Having a little trouble. Okay. So we all make a much larger protein called the amyloid precursor protein. This thing here in blue, and it has a piece in it that's chopped out. And the piece that's chopped out is called amyloid, or beta amyloid, or sometimes it's called A beta. But it's chopped out by these 2 enzymes, one of which is called BASE, and the other one is called gamma secretase. So what happens normally, and we're all doing this right now in our brains, we're making amyloid as I speak, but what happens normally is it disappears. It's cleared from the brain. It either gets dissolved by other enzymes that chop it up into smaller pieces, or else it moves outside of the brain across the blood-brain barrier. So we've been making amyloid our entire lives. In fact, we've been making it since before we were born. But it isn't a problem to us. It's only when we enter this realm where the accumulation begins that it starts to become a problem. The effect of the Swedish mutation is to cause a variant in the protein here such that this enzyme can cut much more readily. It much more easily cleaves this amyloid precursor protein. And as a consequence, beta amyloid or amyloid is produced at a much higher rate. So the individuals that get that rare early onset mutation get the disease because they're making so much more amyloid than we're all making. One of the other things that we learned fairly early on, but has really come to the forefront in terms of therapies over the last 10 years, is that as this amyloid accumulates, it causes inflammation. It triggers inflammation. You can see I've been playing with the keynote. But our understanding of how amyloid is produced also leads to other potential treatment approaches. And the most obvious ones are, if we can stop these enzymes operating, then we can stop the production of amyloid, and by definition, we can stop Alzheimer's disease. It turns out that this has been a very difficult trick to do for biological reasons that I won't go into, but we and most of the pharmaceutical companies have been working on finding ways to switch off these enzymes for 10 or 15 years. Now, there are just some medications that are coming into clinical trials now that do exactly that, and we're all very hopeful that they may work. But they've been a long time coming. The other possible way to deal with Alzheimer's, the other way to think about this amyloid problem, is to find ways to increase its disappearance across the blood-brain barrier. As I mentioned, we normally make amyloid, and some of it gets cleared across. So if we were to take blood samples from all of you, we'd find amyloid in the blood. In the blood, we believe it goes around to the liver and is harmless there. It's destroyed there. But if there was a way to increase the clearance into the blood, that might be an excellent way to get rid of it. And indeed, some of the antibody approaches, some of the vaccines that you've heard about, do exactly that. They produce large numbers of antibodies in the blood, and the amyloid gets pulled across. It's almost a sink effect. It gets pulled across into the blood and is destroyed. So that's another approach. One of the consequences of the Swedish mutation and other mutations like that, which really advanced the field, was the production of chaps like him. He's a transgenic mouse, and he has the normal complement of mouse DNA, but in addition, he has that genetic variant, that human genetic variant that I showed you. So as he ages, he loses his memory. And in many ways, this genetic model recapitulates the pathology and the cognitive impairment that we see in Alzheimer's disease. So these little chaps have become an enormous boon to finding new treatments for Alzheimer's. And again, every pharmaceutical company in the world has mice that have genetic errors, frequently having the Swedish mutation. So why is he in a swimming pool? Well, what he's doing is swimming around trying to find his way out. He doesn't really want to be there. It's not his favored position. And so underneath here, there's a platform, a hidden platform, and he swims around until he finds his way out. And I'll show you some video of that in a little while. But the reason there's a marker on the side here is because mice, like many animals, are very well spatially oriented. And after a few days of being placed in this pool, they learn that the platform is under here. They can't see it because it's milky. This is a milk-colored pool. But he finds out pretty quickly, if he's a normal mouse, that if he swims to the fish, he can find his way onto the platform and shake himself off. But as these mice age, when they have the genetic error, they never get to remember, they never learn that this is the way out. They never associate the fish with the platform. And that's because they develop the pathology that I showed you earlier. There are parts of the brain that are important for coding new information is lost. So when you look in the brains of those mice, they're very similar to what you see in human AD, in human Alzheimer's disease. And here's a comparison, side-by-side comparison. This is one of those senile plaques that I mentioned to you. This is actually from a human brain. And here we have the same thing in a mouse brain. This is the so-called TGAPP3 or transgenic mouse. And you can see they look very similar. And back to my comment about inflammation, what we've actually stained here with is COX-2. COX-2 is the enzyme that gives you a lot of pain if you've got a little bit of arthritis or chronic arthritis. It's the upregulation of COX-2 that causes a lot of pain and discomfort because it releases inflammatory molecules. It produces inflammatory molecules. Same thing happens in Alzheimer's disease. And I say that it's always very fortunate for us that we don't have nerve endings in our brain, because if we did have nerve endings and sensory endings, this would be terribly painful. There's an enormous amount of inflammation. And so again, another strategy, which we could talk more about, has been to develop therapies that can combat this inflammation. Because one thing we know is that the amyloid production and amyloid accumulation leads to inflammation, but just as surely does the inflammation lead to more amyloid production. So there's this vicious cycle that the brain gets into where amyloid leads to inflammation, inflammation leads to amyloid, and that starts this deteriorating process. And there's this idea called the innocent bystander hypothesis, because all around here are neurons. These cells here are actually inflammatory cells, the ones that you see highlighted, but in between are neurons, and those neurons are being bombarded by inflammatory chemicals, inflammatory cytokines, and it's destroying them, it's damaging them, and it's inducing that tau pathology that I showed you earlier. So this is one of the compounds that we've developed at the Roskamp Institute to try and overcome this disorder. It's a repurposing of an old drug. That was our initial strategy to, and I'm sure you're all aware, to develop a drug through the FDA process from scratch is extremely expensive. So one of the approaches that we took was to screen thousands of compounds to see whether any of them might be useful at lowering the production or the accumulation of amyloid. And we took drugs where we already had a good safety profile, so we didn't have to do a lot of the safety testing. And we found, in short, about 300 drugs. If you come to the Institute, and I always say if you happen to be a mouse with Alzheimer's at the Roskamp Institute, you're in the best place, because we have about 300 drugs that can cure you there. The difficulty, of course, is getting one of these drugs into, through the clinical trials, through human clinical trials, and getting it approved for use. So this was one of the drugs that we've taken and looked at extensively, and in fact have moved this all the way through to phase 3. So this drug is a so-called L-type calcium channel blocker. It blocks calcium entry into cells. And as a consequence, in the vasculature, it stops blood vessels contracting. So it dilates blood vessels. And as such, it's an antihypertensive because it keeps your blood vessels open, and the volume of blood has to go into a larger area, and so blood pressure drops. It never made it to the US. It was actually tested here over a decade ago, but it was never approved here as an antihypertensive. It was approved in Europe, and it was approved in Japan. But what I'm going to show you is its role in changing cerebral blood flow, the amount of blood that goes to the brain, what it does to amyloid levels, and when you see this A-beta, think amyloid levels in the plasma, and what it does to amyloid levels in the brain. So this is what happens in those transgenic mice when you give them nilvadipine and look in their blood. You see this increase in amyloid compared to an untreated mouse. This is large increase in amyloid. That amyloid's coming from the brain. So somehow nilvadipine is opening up the blood-brain barrier and allowing the amyloid to move from the brain into the blood, something that we very much want so that it won't accumulate. This is a very interesting effect too. When you look at the blood flow in those transgenic mice, what you see, and this is us looking at different sections of the brain, you see in untreated animals a lot of blue and a lot of greens and not very much red. That's because there's reduced blood flow. And this is exactly what happens in Alzheimer's disease. It's an invariant accompaniment of the disease that with time, blood flow to the brain decreases. And we can get into a little bit more why that happens, but in essence, the amyloid directly attacks blood vessels. And so when we treat with nilvadipine, I think you can see quite clearly that there's a lot more red over here and a lot less blue. That's because the blood flow has increased. And exactly the significance of this, we don't yet know. We don't know if that is in some way increases the amount of amyloid that moves out of the brain, but it certainly increases the amount of blood in the brain, so that's a good possibility. And I'm not showing you here, but we do have there are some human data suggesting exactly the same thing, that nilvadipine can increase blood flow to the brain. But this is probably one of the most important pictures I'll show you. Here's an untreated transgenic animal, and all of these spots you can see are amyloid plaque. So this is the accumulation of amyloid in these animals. And here's a section from, or different sections from the brain of an animal that's been treated. And I think, again, just by eye, you can see there's much less amyloid. And if we were able to bring off this type of trick in humans, this would be a great thing. We could surely delay the disease. And that's what we're hoping to do with this drug. The other important finding is that when the mice get into that pool and look for the way out, and here they are on day 1, and these are different mice, and I'll explain that in a second. By day 9, the transgenic mice are swimming just as fast. So this is the distance that they have to swim, and this is the distance day 1, if you just focus on this sort of mauve color. Day 1, they're swimming this distance. Day 9, they really haven't learnt to swim directly to the platform. They haven't learnt anything. They haven't acquired any new information. However, if you treat them, you see here in red, with nilvadipine, you see this reduction in the length that they have to swim. And that indicates that they are learning that that's the exit, and so they don't have to swim so far. Interestingly enough, these are normal mice here, but if we give normal mice nilvadipine, you can see that they swim even less distance. So even normal mice seem to benefit from the use of nilvadipine, and it seems to increase their cognition. So we do have some preliminary clinical data for this drug in a Japanese cohort. And what you're looking at here is scores on something called the ADAS-Cog. The ADAS-Cog is the FDA gold standard for cognition. It's a scale that measures many aspects of our cognitive faculties. So it measures attention, memory, language function, and so on. And you get a cumulative score. And the higher the score, the worse you are on this scale. So these individuals have been normalized to 0. And here we have a time span in months, so nearly 2 years here. And each of these individuals have been assessed at a couple of points. So this individual didn't make much change and then suddenly got worse. This individual got worse straight away and went up here. Now, these individuals didn't have Alzheimer's to start with. They had this disorder called MCI, or mild cognitive impairment, which is a sort of a pre-Alzheimer phase. But where you see this little asterisk, that means that they've actually developed Alzheimer's. So what we've done here is taken an overall measure of the decline in the whole group. So these were individuals that had MCI, but they also had hypertension. So we gave them an antihypertensive, or they were given an antihypertensive called amlodipine, and you can see they decline. And that's exactly, what you'd see in any cohort of Alzheimer patients or pre-Alzheimer patients, that over this 2-month period, you'd see this type of cognitive decline. So we contrasted that, or you can see the contrast here, with nilvadipine, where nilvadipine seems to plateau the whole group. Only one individual developed Alzheimer's disease, and over this 20-month period, the rest overall sort of plateaued in their cognitive abilities and didn't decline. Again, if we can produce something like this would be a fantastic advance for the disease. Right now, we don't have a drug in the clinic that, available in the clinic that can do anything like this. So the reason we used amlodipine and nilvadipine is that they're very similar in structure. The difference is that amlodipine doesn't cross the blood-brain barrier, so it doesn't get from the blood into the brain. So one of the critical features of nilvadipine is that it does that very well. It gets into the brain. In fact, it accumulates in the brain. And we see this beneficial effect. So as far as nilvadipine is concerned, here's our summary, that it's a well-tolerated antihypertensive. We have a wealth of safety data. In fact, we did a phase 1, phase 2 clinical trial in Ireland on Irish Alzheimer patients, and they tolerated it extremely well, and they didn't drop their blood pressures at all. I'm not sure whether that was the Guinness or just the general robustness of the Irish, but it was an extremely well-tolerated drug. So I don't have any real concerns that we're going to lower people's blood pressure too much. You see these effects of increasing cerebral blood flow in mice and humans, increasing plasma amyloid levels as the amyloid comes out of the brain. It decreases the amount of amyloid in the brain, and it improves cognition in the transgenic model. And as I've mentioned a couple of times now, I've just come back from the launch of this into a phase 3 clinical trial in Europe, 500 patients in 9 countries in Europe. So that's going to be eagerly awaited by us and others. It's going to take, unfortunately, about 2 years or maybe a little bit longer, 2.5 years to complete that study, but very pleased that we're underway with it. There are very, very few compounds in phase 3 clinical trials for Alzheimer's, certainly less than the number of fingers on 2 hands. So we need more advances like this. We need more drugs in phase 3 trials. I will just tell you about a development of that. We have a second compound called ARC31. And basically what we've done is we've taken nilvadipine and we've split it. Nilvadipine actually comes in two halves or 2 parts, and they're sort of mirror images of each other. One of them is blood pressure lowering, the other one isn't. But the one that's not blood pressure lowering still has all the anti-amyloid effects. It still takes amyloid out of the brain. So that tells us that it's not lowering blood pressure per se that's useful with nilvadipine. It's some other property that it has. And we're excited about that because it means we'll be able to raise the dose of ARC31 without lowering people's blood pressure. So we'll be able to give them more and get the anti-amyloid effect. So that's something that we're developing right now. I put this slide back up again to remind me that I'm now going to talk about another compound that we've developed in conjunction with a for-profit organization called Star Scientific. And in this approach, we've really focused very much on inflammation. Some of these slides I'm just about to show you are going to get a little bit more technical, but if one can reduce the inflammatory process in Alzheimer's, one can reduce this vicious cycle, as I mentioned. What we've done here is taken this compound called anatabine, which is very similar in structure to nicotine, and we've done a lot of basic science work on this to understand its properties. Anatabine, like nicotine, comes from the tobacco plant. It seems to be unlike nicotine in a number of important ways. One is it doesn't seem to be addictive at all, according to a study out of Harvard. But it has some other very important effects, some of which mimic nicotine. Here's one of them, which doesn't mimic nicotine, but it's perhaps more pronounced with anatabine. When you see this number up here, RCP006, that's just our internal designation for anatabine. But what we've done here is we've inflamed some cells, and then we've added progressive amounts of anatabine. And what you can see is this reduction in inflammatory cytokines. We're actually measuring something called NF-kappa B. NF-kappa B is a sort of a master controlling protein that controls inflammatory processes in many cells. And so it's very important to see this anti-inflammatory effect of anatabine. And it does something else very interesting too. It cuts down the amount of base messenger RNA. Let me explain what that is. what we've done here is we've increased the amount of inflammation in some human neurons. And what you see here is an increase in the amount of messenger RNA. That's the message that comes from the nucleus to say to the cell, make some more protein. And the protein that it's saying should be made is base, one of those pairs of scissors. So if you make more of that enzyme, that cutting enzyme, you'll make more amyloid. If you make more amyloid, you make more Alzheimer's. And what you can see is when you add anatabine, you really suppress that effect very dramatically. So anatabine has the ability to suppress the pro-inflammatory, but also the pro-Alzheimer effect in human neurons. And here's some confirmation of that. This just shows you that the amyloid levels are going down as we increase the amount of anatabine. There's basically two types of amyloid. This one is a little bit more friendly than this one. This one's called 1:42, and it's the really noxious, really toxic form. This one is less toxic. And some people really think that it's the ratio of 42:40 that determines how much damage gets done in the brain. But either way, with anatabine, you can see this reduction in amyloid production. So this was all preclinical work and very comforting and very interesting. But it was in cell culture. We really wanted to know, could we see the same thing in the brain? And here we have mouse brain, in fact, that has inflammation, a lot of inflammation. And here's a mouse that's been treated with anatabine. And again, you can see this reduction. So that the idea is that this anatabine can very easily get into the brain and suppress inflammation and suppress the production of amyloid. So of course, we and others are thinking that this might be something that we can, that might be beneficial in Alzheimer's. I will just explain one thing though at this point, and that is this is being developed as a nutraceutical, not as a drug. And there's a big difference. There's a huge difference as far as the FDA is concerned. There's a big difference in terms of regulation, and there's a big difference in terms of cost. To give you an example, just filing what is called an investigational new drug application for nilvadipine cost us at least $10 million. And that was just to produce the paperwork and some studies. One doesn't need an IND with a nutraceutical, so you can get into clinical trials or clinical use or just general use much more rapidly. However, the FDA has very, very strict rules about what is a nutraceutical and what isn't. And if you try and modify diseases, then the FDA regards that compound as a drug, essentially. So that we can't advance this as we normally would as a drug, but nevertheless, these are very interesting preclinical findings, and this is a compound that's actually available already on the internet and elsewhere. But this I think you might find interesting. Here are those two, here are mice, one treated with anatabine, one treated with placebo. And I can tell you, because you probably can't see it, that the hidden platform is about there. It's about there in both cases. And these are transgenic mice. So these are mice that by the time they were asked to, by the time we put them in this test, maybe we did ask them, we're very kind to our mice at the Institute. By the time they agreed to do this test, they had a lot of Alzheimer's pathology. They were quite old, in fact, and had a lot of amyloid pathology. And so you can see that but this is on day one as well. So day one, it's really rather exploratory. But you can see they're swimming around the perimeter, and this is fairly typical, and it'll take them quite a while to actually, just by chance, wander across and find the platform. And again, you can't see from where you are, but there are markings on the wall indicating that, to give them some signposts that that's where the platform is. But look what happens on day 5. Here's the anatabine-treated mouse. This mouse has now been treated for 10 days with anatabine. So 5 days before it was asked to do this study, but this one, which is also an Alzheimer's mouse, has had no treatment or placebo treatment. And you can see this animal has gone straight to the platform very quickly in a very short space of time. It's really quite remarkable. So, and this one is not doing a whole lot better than he did on day 1, which is very typical of the Alzheimer's mouse, unfortunately. So this is very encouraging to us, and we're very interested in this effect, and it's something that we're exploring very very vehemently. And as I mentioned, in addition, this is something that's already available as a nutraceutical or food supplement. So the last little section, I'm going to talk about what can we do to offset our risk for Alzheimer's disease. And I think there's quite a lot. There's probably a lot more than you think. One of the things that we now know is that for whatever reason, these disorders all confer an independent risk for Alzheimer's disease. Well, what do I mean by that? Well, if you have hypertension and you have a small stroke, that can enhance and exaggerate Alzheimer's disease. So that's a clear link between pathology and making Alzheimer's worse. But for reasons that are still not clear, just having diabetes or just having hypertension without a stroke or certain cardiac diseases or high cholesterol or high triglycerides, all of those things are independent risk factors for Alzheimer's. And I put the word aggressive in here because I think our views about how well these disorders have to be controlled to mitigate risk for Alzheimer's is changing. We have to actually control them much more than we thought we needed to. So some of the recommendations that are out there for reducing blood pressure into what we consider a normal range might not go quite far enough to reduce the risk for Alzheimer's disease. But this is something that your doctor or your doctors can do. These are things that we really need to take care of. Having atrial fibrillation with pulse rates of 120 just isn't good enough. In not many years ago, that was considered okay and people would let that pass. But actually we know that that's a particular risk for Alzheimer's disease and will induce the disorder. So we should consider diet as well. And this is an area that we're learning more and more about. But my conclusions are that it's really a combination of dietary factors that might be protective if you look at the literature rather than any one particular component. But there's been quite a lot of talk about the so-called Mediterranean diet, which I've detailed here a little bit, which consists of high intake of vegetables and fruits and cereals and a lot of olive oil, which provides unsaturated fatty acids. Relatively high intake of fish, but low intake of saturated fatty acids, particularly those in meat and poultry and dairy products. And here's the piece I like, this regular but moderate amount of alcohol. So we talk a little bit more about that. But there have been quite a few studies now that show this type of thing, that if you look over a 10-year period, and I believe this was a study done in New York, on a large number of subjects, so we're not just talking about studies that are off the cuff or not reproducible. This is a large number. What you see here is a so-called survival curve. You start off with 100% of the population. 10 years in, if you look at the green, about 70% of them still don't have Alzheimer's. About 30% do. But also 10 years in here, approaching 50% of the ones in red have Alzheimer's. And so what's the difference? Well, the difference is the ones in red had a very low adherence to the Mediterranean diet or Mediterranean type diet, and the ones in green had a very high adherence. So just going from this high adherence to a low adherence, there may be a 20% change, 20% difference in risk for Alzheimer's. And if you actually run it back in years and say, well, look at this cutoff here at 70%, that brings us down to about 6.5 years, and 70 there brings us down to maybe 8 or 9. So maybe there's 2 years to be gained from changing diet. So it's very interesting, I think, that it can be modified in that way. So we probably all know an awful lot about good and bad fatty acids and the ratio of these two. And the idea is that the so-called omega-6 fatty acids are bad news and produce not only cardiovascular disease and put us at risk for diabetes, but certainly there's good evidence they put us at risk for Alzheimer's too. And there's a lot of talk about keeping this ratio of the bad ones, the omega-6 to omega-3, and maybe in the order of 4 to 1. Now, the average American diet is more in the order of 10 to 1. So So there's quite a bit of work to do with many of us. Now, the omega-6 fatty acids are very strongly associated with constriction of blood vessels and inflammation. And we know in the animal models that when we inhibit arachidonic acid, for instance, we can stop inflammation, we can reduce amyloid production. So again, I just want to point out one thing about this type of study that recurs over and over again. What you tend to see in the literature is when people look at cross-sectional studies, they see benefit. They see benefit with certain vitamins, they see benefit with estrogens, for instance, they see benefit with some of these fish oils. But however, when they look on a forward-going basis, in other words, if we were to take all of us now and say, okay, half of us are going to go onto this diet and the other half of us won't, very often those studies don't pan out very well. We don't see the beneficial effect. This happened with non-steroidal anti-inflammatory agents that we and others were looking at. And I think the reason for that is you have to get in very early. I mean, typically what happens when people do forward-looking studies, they can't wait forever. So they usually pick people that are 75 or thereabouts and look at them going forward over 5 to 10 years. But actually what we need to do is wind the clock back much earlier. I think what these studies are telling us is that changing our risk for Alzheimer's has to occur a lot earlier than we think, maybe in our 40s and 50s. And that's when we should really be thinking about our diet. And changing our diet much later on might not have anywhere near as much effect. So supplements, I'll just touch on quickly. These are good examples of what I've just talked about. In cross-sectional studies, both ginkgo biloba and vitamin E and C both look very encouraging. But in going forward studies, in randomized control studies, those effects have not been replicated either for ginkgo or for vitamin E and C. Folate is one of those few vitamins where there are clear benefits of having good and maybe even slightly elevated folate levels, again in cross-sectional studies. But even taking very high doses in individuals that already had Alzheimer's disease, there was no change. So again, I think the message is do this type of thing early, balance your diet early, make sure that you're not vitamin or folate deficient early. Curcumin is something you've probably heard quite a bit about. The incidence of Alzheimer's disease in at least parts of India is lower than it is in the US. And the thought is that that's to do with curcumin that goes in the curry. And we, in our own animal studies and many others, have shown that it is indeed quite a potent anti-inflammatory agent and consequently reduces amyloid buildup. Back to something that will resonate, and that is alcohol intake. And there have been innumerable studies now that suggest that a modest amount of alcohol intake is protective for Alzheimer's. So if you take individuals that don't drink at all, their risk is here. Individuals that drink modestly have a reduced risk. However, if you drink excessively, your risk will come back up again and actually exceed not drinking at all. But so how much is enough? How much is moderate? Well, there have been a couple of studies suggest that maybe as many as 3 drinks a day, 3 of glasses of wine, is still reduces your risk for AD. I'm not suggesting that you go out and have 3 drinks. I'm just reporting the data. So. But here is one that's close to my own heart, and that's resveratrol, which is was originally examined because of the effects of drinking red wine. Resveratrol occurs in the skin of red grapes, but actually there's very little of it in red grapes. And so supplements, resveratrol supplements are being produced and are available. And that's actually been examined in phase 2 clinical trials now. Here's something that I think we really do need to take seriously. Physical exercise. Study after study now is suggesting that physical exercise has a big impact on the brain. This was a particularly impressive study with 1,500 Finnish individuals were advised to take physical activity at least twice a week through middle age, and their risk of developing Alzheimer's was cut in half. These are really dramatic effects. Again, we see the same thing in the animals, that the greater the blood flow to the brain, as a consequence of exercise, the greater the reduction in risk for development of AD and accumulation. I don't know if you can see this clearly down the bottom, but the one molecule that I haven't talked about today, and maybe we can in the discussion, is this thing called ApoE. ApoE is a genetic risk factor for the disease. We all have ApoE, and if we have 1 or 2 copies of E4, a particular variant, we're much more at risk for the disease and much likely to get it earlier. And it was interesting to know that the most protection they saw in this Finnish study was conferred on individuals that had ApoE4. And that's particularly important because some of the clinical trials that have been run suggest that there may be beneficial effects of certain drugs, but E4 individuals are the most resistant to that. So this is particularly encouraging. Okay, and then the last point is we need to use our brains. We know that individuals that come to the clinic, for instance, that have high educational levels tend to come in much later, and they tend to find ways around the disorder and cognitive strategies that, that individuals with less education are unable to muster. So, but there are a lot of outstanding questions about that. How exactly should we use our brains? How often should we do it? What exactly should we be doing? So this is an area of research that's very much open to further work. All right, so here's the summary. This is what you need to do to avoid Alzheimer's. Be careful of your general health, particularly the cardiovascular system, diabetes, the heart. What's your diet? And I think this is a developing area. We're going to learn a lot more about exactly which diets we should be on and which ones we shouldn't be on. But in many ways, they're looking as if they're the same ones that we should be on to avoid heart disease and avoid hypertension. Exercise seems to be very important. Drink alcohol modestly, use your brain. And I think going forward, what we're going to see is not one magic bullet treatment for Alzheimer's disease. I know that the press is always looking for that, and maybe we're all collectively looking for that. I really don't think that's what's going to happen. What's going to happen is we're going to nibble away at this disease, or maybe make significant advances, but from different angles. I think we'll suppress the inflammation, we'll lower amyloid production, we'll move amyloid out of the brain, we'll support tau, we'll do all those things simultaneously. And so I think you're going to see this cocktail concept being introduced much more than what I believe is a little bit unrealistic right now, that we'll have this super drug that's suddenly going to emerge and cure all our woes. I don't think that's the history of medicine, and I don't think that's what's going to happen here. But I personally have said for a long time, it's not if we cure Alzheimer's disease, it's when, and I really wholeheartedly believe that. That's why I'm at work every day. And finally, please support Alzheimer's disease research, because without your support, this work and work like it can't go on anywhere in the world. And finally, let me just acknowledge my support, which comes from Bob and Diane Roskamp, who are really the founders of the Institute. We're eternally grateful for them and their enormous support over the years. But we're also funded by these peer-reviewed groups, the National Institute of Health, Department of Defense, Veterans Administration, National Alzheimer's Association, and others, and thanks to Star Scientific for collaboration on the inatabine work. And finally, here's just a few of the scientists in the lab, I didn't have room for all of them in one list, who've been working on this and other projects. Thank you for your attention.
For those of us that don't have 2 years to wait, where can I beg, buy, borrow, steal this nilvedipine, and the hell with the side effects? Who cares?
Well, I admire your spirit, and you're not alone in that. But unfortunately, the FDA doesn't agree with that approach. So actually, it's only available in Europe and Japan. So it's unfortunately difficult to get hold of, and I can't say any more than that. But it's horrible as it is. We have to wait for the results of clinical trials, and that's the strategy that we all have in place, is that's what we have to abide by.
You didn't mention heredity as number one. And the second question I have is, you mentioned aerating the brain, in other words, getting more blood to the brain, and CPAP, which my husband and I both use. Do you think that has any benefits?
Okay, so the first question on genetics. So I showed you those very rare early onset families. The majority of us, and in those families, 50% of the offspring develop Alzheimer's. So it's kind of a flip of the coin. If you get the disease, everybody has 2 chromosomes and 2 amyloid genes, and so you pass on one or the other to your offspring. And it's a flip of the coin. And if you're unlucky enough to get the one with the genetic variant in it, you get the disease. Now, fortunately, that's very rare. For most of us, more subtle genetic risks are at play. So ApoE and ApoE4 is actually inherited in the same way. That will increase your risk, but not so dramatically. In other words, even if you have an ApoE4 gene variant, you won't necessarily get the disease at 65. There's just much more of a tendency to. And certainly if you take a large group, then as a group, they'll get the disease earlier. We did a study in Miami where we looked at individuals who had E4 and individuals that didn't. And the individuals that had E4 got the disease on average in their mid to late 60s, where the individuals that didn't get the disease until 70 or above. So it makes a difference. And I always think that's a pretty cruel time period. That's when people retire. And if you happen to retire and you're at risk with ApoE, then you're immediately going to start to sink into this disorder. The other question about oxygenating the brain, it's very difficult to force the brain to take oxygen when it doesn't want it. We have all sorts of mechanisms in place to stop that happening. I think that's one of the reasons why exercise might be an interesting way to go, because actually under conditions of mental exercise or conditions of physical exercise, that is a natural thing for the brain to do. It does take in more blood and it does take in more oxygen. But just adding oxygen to the blood, I'm not sure whether that has a huge impact on the brain or not. It's an interesting idea, but the broader idea is, yes, do we want more oxygen in the brain? Probably. Certainly in Alzheimer's disease, that's one of the things that's going wrong. But there's a big debate in the field. In the field, many people think that the reason that there's less oxygen uptake and less glucose uptake in the brain is because the neurons are dying. It's not because they can't access it, it's because they're actually dying and they don't want it. Whereas there's another group that think, actually, the neurons do want it, but they can't get it because there's a lot of damage to the blood vessels. So that debate's not really resolved. Anybody?
How far the studies on anatobin gone so far? You say it's available. You say it's available.
Yes.
And what studies have been done on it, and how far have they advanced?
Right. So the studies under anatobin was primarily developed as a smoking cessation tool. But we currently have a study underway looking at inflammatory markers in a group of individuals that have high inflammatory markers. This is in Flint, Michigan. So that's the most advanced study right now. There aren't any studies at this point in Alzheimer's disease. Lady there. Oh, yep, that's good.
Yeah, my question is kind of like the gentleman's up front and the gentleman over here. I mean, there are people suffering every single day, and you see them, and you go to nursing homes, and you see them suffering. But are these nutrapharmaceuticals being marketed to people that have the disease now, or are they available on store shelves at GNC and these other places? Where do you get the product?
So the anatobin is actually available at GNC. It's just become available at GNC. It's available online. And I have to, again, caution, we don't know that that's going to work in Alzheimer's. I've showed you all the preclinical data. I've showed you that it looks like an anti-inflammatory molecule preclinically, and I've showed you that in the animal models it looks good. But sometimes it's a big jump between mice and men. Frequently it is. But that is available. as you know, and the reason why I get up in the morning is because we've only got 3 drugs that are approved by the FDA for Alzheimer's disease, and none of them work very well. So my mantra is we need more of this type of thing. We need more drugs in clinical trials. I mean, I think it's heartbreaking that in phase 3 trials, we just have literally a handful of drugs. So we need to do more, and I think there needs to be more emphasis on translating the science into drugs, not just the science for science sake. I think we need to do more translational work, and we need to be more ambitious about getting drugs into clinical trials. I'll just say one point about that. The drug companies don't like Alzheimer's very much because it's so expensive to run a clinical trial. It's not like, if we were talking about cancer, I could literally tell you about hundreds and thousands of drugs entering clinical trials a year, or being considered for clinical trials. In Alzheimer's disease, you typically have to run the study for 18 months. That's a very expensive study to do, and the drug companies don't like that, and there have been some spectacular failures as well in the field. So some of the companies are just closing down their dementia R&D units. So that's why I have some hope for nutraceutical food supplements. That's a quicker way to go. I think a lot more people are interested in that, and I think there are compounds that are very biologically active.
When someone their mother had it, now they have been diagnosed early, 50-something, and yet they've exercised, drank wine, had a good diet, had a very good education, and they aren't going just down in the toilet. There's no hope, I guess, for people that inherited it.
Well, if you inherit one of those rare early onset mutations, those mutations will overcome almost anything. I mean, they're very powerful. It's a very heavy dose of amyloid to get. I will tell you one story, though. My wife is in the audience, actually. She probably remembers this. In the early days, I used to go and visit early onset Alzheimer families up and down England. That's how we got started in this 20-odd years ago. And there was one family where everybody would get the disease in their 50s, in their early 50s. And I knew from the genetic data that one individual looked very much like he had the genetic error. So I was visiting him, and I couldn't quite understand why he hadn't succumbed to the disease. He was 56 and still hadn't succumbed. So I visited him at his apartment, and all over his apartment were puzzles. He did these puzzles, and moreover, he'd been a parts manager in a car, in a garage, and he had remembered the different parts of Jaguar cars by heart. So he'd been working on his, so people would come in and say, I want a carburetor. And he'd say, oh, you mean part BT7504? And so he'd, use it or lose it. But no, the reality is that for those very early onset cases, that's a heavy, heavy burden and very difficult to deal with. But for the most of us, where genetic influences do play a role, but they're not overwhelming, I think using our minds and taking some of the advice I've suggested here is helpful.
What is the difference in Alzheimer's and dementia?
Okay, so it's just a definitional thing. Alzheimer's is a form of dementia. Dementia means that you lose your memory, you lose some other cognitive faculty, so two things, and in addition, your social or work environments are impacted. So your memory's gone, maybe you're losing language function, and it's impacting your work, your environment. So it's just and there are many, many causes of dementia. It just so happens that Alzheimer's is now the most common one. Maybe I'll take one more question. Lady there.
Oh, wait, hang on, I'm sorry.
Two more questions. Yep, sorry.
Do you know of usage of coconut oil and MCT?
Right. So there's been a lot of interest in coconut oils. and other diets like that. And these are related to the idea that the brain can metabolize, can use certain other substrates other than glucose. And it really has to do with the induction of a diabetic type state, a glycemic state. It's interesting, again, in the animal work, there's some suggestion that it might be helpful. I don't think we have any proof from clinical trials yet, but I know there's a lot of interest in pursuing that. Yeah. Last question.
Okay. What about the nasal insulin?
Yeah.
What's going on with that?
Yeah, so the links between Alzheimer's and diabetes are quite profound. And the lady's referring to the administration of insulin into the brain directly through the nasal passages. The olfactory nerve goes directly into the brain, and interestingly enough, insulin can sort of travel probably around the outside of the nerves and find its way into the brain rather quickly. Again, in animal models, it's beneficial. It reduces inflammation, it increases normal metabolism, and it stops amyloid production. So there have been some very small phase 2 clinical trials up in Seattle suggesting some benefit. So that's something that's going to be very closely watched too. Yeah. Again, thank you very much.